introduction, and yeah, so I'm just want to go first to the content of this presentation and the content I will show you in the next slides and in the next about half an hour. So first, of course, I will give you a short introduction in the field. So this will be, of course, a very short introduction because most of you are, I guess, are really experts in the field. And after this introduction and this introduction into the problem that what we are dealing with, free circulating nucleic acid in blood, I show you the development that we have done during the last years here in our group with respect to CCF DNA. And first of all, I show you the performance and features of the new PAXgene Blood CCF DNA tube as part of the system that we will introduce shortly in the market. And in the next part, I will show you the features of the QIAsymphony PAXgene Blood CCF procedure, which is linked to the tube, and this together is the system that we introduce here. And finally, I give you a summary about all these isolation procedures and stabilization procedures into the context of the whole CCF and liquid biopsy campaign here at QIAGEN. So in the next slide, this is a very nice picture I often show from a Nature publication, where we see what the CCF is about, the CCF DNA is about, and all the other CCF nucleic acids, and what are the issues that we have to deal with if we develop systems that are used to stabilize these free circulating nucleic acids and isolate them from plasma or other body fluids. So the CCF DNA normally is strongly fragmented, and most of the fragments that we have in blood or in other body fluids are far below 500 base pairs, mostly much more shortened than 100 base pairs. But we also see other fragments that are much longer, and they come mostly from necrotic processes as shown in the picture below. So we have different processes, as you know. So we have necrotic processes, which is more an undirected process, and other processes like apoptosis or secretion of DNA into the bloodstream that are more regulated processes here. And yeah, so what we also have to deal with is that the CCF DNA in plasma, serum, or urine has also very low concentration, and this is one of the most important issues that we have to handle with during the isolation and stabilization of these very important nucleic acids. And in the next slide, I just want to show you very simply what is the issue and why we developed a new system for stabilization of CCF DNA. And what you see here is a graph from the Agilent bioanalyzer where we see the CCF DNA isolated from EDTA plasma, so from blood collected in EDTA tubes directly after the blood collection. The plasma was separated from the blood cells, and the CCF DNA was isolated. And what you see here is a very small amount of CCF DNA between the two marker peaks in the Agilent. And this is directly after blood draw, and if you have to transport or store the blood for a certain time, then strange things happen, and this is shown in this slide here. So in the next part of the slide, so strong apoptosis occurred with the blood cells, with the white blood cells, and release huge amounts of genomic DNA, and these huge amounts of genomic DNA, of course, dilute the original CCF DNA, and this is in the case and in a way that you most likely do not find your original CCF DNA anymore in this huge background. And therefore, our idea was to develop a system that prevents the cells, the white blood cells, from lysis, and also facilitate a streamlined workflow for CCF isolation to have all the benefits on this complete system here. And in this next slide of my, the next few slides in my talk, I show you the first part of the system, the PAXgene Blood CCF DNA tube, which will be released this year. So in this slide, we see an overview in table format for the blood collections tube that are mainly used in the field on the left part of this table. So the EDTA blood tubes or the STRAC CCF, the STRAC cell-free DNA blood collection tube, and our new tube that we developed here at PreAnalytiX. And in the first line, you see that, of course, is an important point if you are interested in molecules, cfDNA molecules that are very rare in your blood, that you need a certain volume of blood and a certain volume of plasma to deal with. And you see that all the three systems collect 10 ml of blood, and you get more than 4 ml of plasma out of it. This is the same for all three systems shown here. But in the next row, you see that the tube material itself is different. So the blood, the normal EDTA blood tubes are of course from plastic, made from plastic, but the STRAC tube is made from glass, and this could of course bring with some issues with it. So because the glass can break, and you can have issues with during transport, but also during centrifugation if you prepare the plasma. And this really happens if we know from several field testers and customers in the field. So our new tube will be made, of course, of plastic as well. So then, and as I showed you in the last slides, the centrifugation at blood collection site is needed for the EDTA blood, because if you do not do this, if you do not separate immediately the cells from the plasma, then you get this huge amount of gDNA contamination in your plasma. This can only be avoided if you directly separate the plasma by centrifugation, or if you have a very extensive cooling change to prevent this. And this is, of course, not what you normally have or can make during your studies. So the STRAC tube for the STRAC tube, you do not have to do this, because in the STRAC tube you have a stabilization solution in it that prevents the white blood cells from lysis, and therefore you do not get this huge gDNA contamination. In our new tube, we also have a blood collection blood stabilization solution that is completely different from the STRAC one, but the effect is the same. So these white blood cells are prevented from apoptosis. So for these tubes, you do not need this centrifugation, and you can store and transport the blood for up to 7 days at room temperature. This is, of course, as I showed, not possible for the EDTA tube. So what is the difference then between the STRAC tube and our tube? So our tube does not contain any cross-linking reagents, so you have no modifications of the DNA and all the other biomolecules in your tube. So this is different for the STRAC tube, and this is shown in different patterns mentioned on the bottom of this slide. So our stabilization solution does not contain any cross-linking reagents. And what is also different to the STRAC tube and to the EDTA tube as well, that we standardize the complete workflow. So we will not sell a tube only, but a complete system. So we looked at the workflow for plasma generation, established protocols here that ensure that you have clean plasma out of our tubes, and then directly have developed dedicated procedure for isolating ccfDNA from this plasma in an automated way, so with the QIAsymphony as an automated instrument behind this. So you get everything from one supplier and have a complete workflow from the sample to the ccfDNA in the end. So in the next slide, I will show you some data that we generated with our new tube in comparison to other tubes. So first point and first aspect I want to talk about is hemolysis. So we, with our tube, also prevent the red blood cells from lysis, and this is shown in this picture here. So you see over 10 days that our tube here shown in green, and the EDTA tube in blue, that the hemoglobin is very strongly distributed in the EDTA tube over time. So you get so the red blood cells lyse, and our tube stays more or less constant for with respect to hemoglobin and hemolysis. Of course, the red blood cells normally do not contain any genomic DNA, and therefore you can of course say, so what is about hemolysis with our systems? But in the next slide, I show you an example for how hemolysis could look like in other tubes, and you can imagine that with a strong hemolysis, you also can get issues to isolate the plasma from the blood cell fraction. So if you have a strong hemolysis, then of course you can probably not get as much plasma out of the tube without cross-contamination of white blood cells if you have a strong hemolysis background. But of course, we looked also in the DNA out of the tube. So first we looked here, and that is shown here in the genomic DNA from the whole blood, and where you can see really in this picture that the white blood cells go in apoptosis. So on the left part of this G picture, you see EDTA blood in EDTA tubes and blood in our new PAXgene tubes. And the DNA, the genomic DNA of the whole blood was isolated directly after blood draw, and you see this nice strong genomic band. But after 10 days with this blood from the same donors, you see that these apoptotic bands for the EDTA plasma occurs and shown here, and with our new tube you do not see any of these apoptotic bands. But this, as I said, of course, is genomic DNA from whole blood. But we, of course, are more interested in the ccfDNA, so the free circulating DNA, and this, what happens there is shown in the next slide. So this is a picture I showed you again. So you see you see the EDTA blood directly after blood draw, no apoptosis, and after 10 days, strong apoptosis here in this Agilent picture. And if you do the same analysis with directly after draw and 10 days after draw with the PAXgene tube, you see, and this is shown here now, you see that there's also no apoptosis with respect to CCFDNA and from plasma shown in the Agilent picture here. And you can, of course, also look at the CCFDNA with PCR, for example, and this is shown in this slide here. So we have here a qPCR assay for the 18S rDNA, and we isolated the ccfDNA from plasma after different time points, shown on the x-axis here. And the EDTA blood, you see a strong increase of the 18S rDNA from the plasma. But you see in comparison to this, for the PAXgene ccfDNA, no increase of 18S ccfDNA up to 10 days. So this is a PCR, and of course, many of you probably use the new NGS technologies here, and we also tested our tube with this technology together with a partner, LifeCodex here in Germany. It's an NIPT company, and what they have done here in a research study with 18 blood samples from pregnant women collected in parallel in the PAXgene tube and in the STRAC tube. And after blood collection, the samples were sent to LifeCodex, and they isolated the plasma from these tubes. And in the first step, they then isolated the ccfDNA from both plasma types and analyzed via a PCR assay the fetal fraction, so the portion of fetal DNA in the background of maternal DNA in the ccfDNA, and their criterion, the QC criterion is that this fetal fraction have to be at least 4%. And if you look at the pictures at the bottom of this slide, on the left you see this assay, so it's called quantification. It's a PCR assay, and you see that between the STRAC tubes here in gray and our new tubes in green, and that there is no difference in the fetal fraction between both plasma types. And if you go more to the right, to the two pictures on the right, you see also in the NGS workflow that you have a very absolutely comparable total reads in the NGS workflow, and the mapped and unique reads are also not different between the two tubes. So with their PreAnalytiX assay, they analyzed these 18 samples for trisomy 13, 18, and 21, and they are all concordant. So as they use at the moment the STRAC tube, the PAXgene tube was completely concordant to the current method they use. So overall, we can summarize that for ccfDNA, our tube is absolutely comparable to the STRAC tube, and better with respect to genomic DNA contamination to the EDTA tube, and has several features that are different with respect to safety, and this is shown here now. So we have plastic material with the tube, so this of course helps to avoid tube breakage. We have at the top these safety BD Hemoguard closures, so this of course is also different compared to, for example, to the STRAC tube. This avoids that the blood in the tube, if you open the tube, contaminate your workbench or even your other samples. Then, as I showed you, we prevent hemolysis, so this helps you that you can have a very safe plasma separation, so that you get maximum plasma yield from your blood. And then finally, we have no cross-linking reagents in our stabilization solution, and this is of course positive with respect to CCF preparation and assay performance. Okay, so this was the first part of the system, the PAXgene Blood ccfDNA tube, and now I come to the second part of the system, so the QIAsymphony PAXgene Blood CCF procedure, also available this year. So to start with this, I just summarized here or give an overview about the workflow from the patient to the results here. So it of course starts with the blood collection with the currently available tubes. So for example, the STRAC or an EDTA tube, then a transport, plasma generation, CCF isolation, for example, via the QIAsymphony instrument, and then the analytical assays, NGS, PCR, and the results. And QIAGEN already introduced last year a kit on the QIAsymphony that's called QIAsymphony Circulating Nucleic Acid Kit, and this kit is a generic CCF isolation kit that works with STRAC or EDTA plasma, and you can isolate the CCF from these tubes here. To distinguish this, I show you now our new kit and our new system, and this is a little bit different approach here. So this system is completely closed, so we have our own tube now with a stabilization solution in it. You have a workflow for transportation up to 7 days at room temperature, a protocol for plasma separation here, and then finally a dedicated kit and a dedicated protocol for the QIAsymphony to isolate the ccfDNA out of it. So complete system from one supplier and with a dedicated workflow for everything. So compared to the generic system from QIAGEN that works with all kinds of plasma that you isolate. Okay, coming to this, I show you some of the workflow in a little bit more detail. So what we offer here, we offer two kinds of protocols for the QIAsymphony, all running with the same kit. These two protocols address two different plasma volumes, 4.8 and 2.4 ml of plasma can be used, and then you have proteinase K digestion, add metagenetic beads and binding buffer, then finally the plasma and do the binding steps, And if you use this large protocol for 4.8 ml of plasma, then you only separate the beads from this first binding step and use the same beads with the bound ccfDNA from 2.4 ml of plasma again with another 2.4 ml of plasma, so that you have overall 4.8 ml of plasma, and go then to this process again, and finally have 3 wash steps and elution, and elute your DNA with the option of 3 different elution volumes. So in summary and in comparison to the QIAGEN QIAsymphony circulating nucleic acid kit and the differences compared to this kit, so with our new PAXgene QIAsymphony kit, we have a dedicated isolation technology for the plasma generated from the PAXgene Blood ccfDNA tube, so optimized binding chemistry for the plasma from the new tube, and optimized protocol steps to get the maximum yield out of our plasma. And then we have 2 protocol lines, so a standard protocol line, so more focused on small cccfDNA fragments, and then another protocol line focused on large and small fragments, so that you can really address all kinds of cccfDNA with one kit and different protocols dedicated for plasma generated from the PAXgene Blood ccfDNA tube. And finally, we have the option to have 3 different elution volumes. So now coming to some data, comparing these 2 QIAsymphony ccfDNA isolation procedures. This is shown here in this graph. So on the left part, you see the isolation with with our new PAXgene Blood QIAsymphony kit, and on the right part you see blood cccfDNA isolated from plasma of the same donors from the PAXgene Blood ccfDNA tube with the generic kit, so the QIAsymphony circulating DNA kit. And if you look more closely to these bars here, you see that you have slight advantage with respect to yield. So we should see copy numbers on the y-axis here for the dedicated PAXgene QIAsymphony kit compared to the generic kit. And this is of course not surprising, because for the generic kit you have different kinds of plasma samples, plasma types coming in, and you have of course a generic idea behind this kit. And with our kit we have really a dedicated solution for the plasma coming from our new tube, so that we have a really complete system. Okay, so finally I'd like to summarize what I showed you in the last slides here. So overall, we can, for QIAGEN, can now offer complete solutions for all kinds of ccfDNA from plasma. So, and this is shown in this nice pictures here. So there's also again this complete workflow from patient to clinical results, and we focus on the sample and the pre-analytic workflows, so including plasma blood collection, logistics, plasma separation, and ccfDNA extraction. And if you look to the lower part of this picture, you see if you use our new tube, you can isolate it by our new QIAsymphony kit in the middle, or also, did not mention this, via manual process via the QIAGEN circulating nucleic acid kit. So this is a manual kit. Or if you have EDTA or STRAC plasma still, then you can use this generic QIAsymphony circulating nucleic acid kit. So all plasma types that are in the field at the moment can be addressed by solutions from QIAGEN. Okay, so this was my last slide, and I want to thank you for all your interest here.